This is Biochemistry Unfolded, From Atoms to Algorithms, a weekly podcast on the topic of computational biochemistry and biophysics. Welcome to the inaugural episode of my new biochemistry podcast, Biochemistry Unfolded, From Atoms to Algorithms. My name is Dr. Derek Cashman, also known by the nickname Dr. Cash, and I am currently faculty in the chemistry department at Tennessee Technological University in Cookeville, Tennessee. This is a weekly podcast that I'll be producing in 2025, covering current topics and perspectives in the field of computational biochemistry and biophysics. If you are a student, postdoc, faculty, researcher, or just someone who has an interest in biochemistry, I hope you'll find this content and podcast very interesting and informative. If you have any ideas for content that you'd like to see, please leave them in the comments below. And also be sure to hit the like and subscribe buttons so you can stay informed and updated on future episodes. To begin, I thought I'd provide some background into my journey as a scientist and how I arrived at my current position today. I think most of us remember many of the teachers and mentors from our education that inspired us in our educational journey, yourselves, and in your careers. This is no different in my own experience. I traced my interest in life sciences to my middle school life science teacher, Mr. Shelton. He's now Dr. Shelton, having received his PhD about a year after I was in his class. Congratulations. His way of presenting, and this was middle school, and his way of presenting the course material was very engaging, and it inspired me to learn more. The same year, we had a family friend that we met through church, Dr. Jack Haar, who was a professor in the anatomy department at Virginia Commonwealth University. And he invited us to his laboratory and to kind of see what he does. And so that was really cool. And it was really my first campus visit to the university, where 20 years later I'd actually get my PhD. In a different department, but still. I went on to high school. I probably got a little distracted there. Thought it might be interested in engineering initially, so I took some courses in mechanical drawing and architecture. Didn't have a very inspiring teacher, and I kind of lost interest. Two of the courses in high school that resonated the most with me were physics and Pascal programming, even though my math background was fairly weak. I did have a strong interest in computers, and I was even accepted into an undergraduate program in computer science, but I wasn't too sure if I wanted to do that. I had to buy a computer, and so I went on and enrolled as a biology major instead, and that was at Old Dominion University. And it was here that I met my second major inspiring teacher of my academic career. That's Professor Nancy Wade in the Department of Biology. Nancy Wade taught the first year general biology course, and this was particularly known, maybe infamous, for being the primary weeder course for all the pre-med students trying to get into med school. Half the class failed or got a D, but the other half, we did learn our biology. She only probably gave about 7 or so A's, and I was happy enough to get a B, and that was out of a class of 300. She also gave a lot of useful advice, like stopping by to smell the roses, enjoy life, take a break from the daily rat race we're all engaged in. Remember, she drove an orange Volkswagen Beetle with the license plate Tunebug. Yeah, she was a trip. Probably one of the most memorable professors at ODU that even most students will remember. 4 years later, I graduated ODU with my BS in biology. I had a minor in chemistry in 1995. I was also probably one of the few bio majors that actually took coursework in programming. And with the rise of the internet in the mid-90s, my first job post-graduation was actually working for a small company in Eastern Virginia. And they were contracted to work on the website for CBS television. So I guess then I can say I kind of sort of have a background in TV, right? All right. And it was about this time I really decided what I wanted to do was chemistry, not biology. Well, I mean, a little bit of both, but I found the material in chemistry more interesting. I was really interested in drug discovery. But in order to do that, I had to take calculus prior to graduate school. So I always struggled with math courses. But fortunately, this time I had a good teacher. He was only adjunct faculty. He actually had a regular job as a naval contractor. But when you know what you want and what you need to get there, you can definitely get it done. And that's what I did. And so I did well in math. I did my calculus. I finished. I went on to graduate school in the Department of Medicinal Chemistry at Virginia Commonwealth University. And I studied under Dr. Glenn Kellogg. And I also learned QSAR from the greatest, Dr. Keir, Dr. Almont Keir, and Dr. Don Abraham. My PhD research involved drug discovery in small molecule anti-cancer agents such as doxorubicin and other aminoglycoside antibiotics. We used a program called HINT, which is short for hydropathic interactions. And this is a log P-based scoring function that predicts the binding free energy of small molecules and of proteins and nucleic acids. So my dissertation is entitled Computational Models for Targeting Nucleic Acids, Estimating Changes in Free Energy for Drugs in Complex with DNA, RNA, and multiple sequences of DNA. And this was completed in 2003. Then I went on to do postdoctoral research, initially at the University of Louisville, working for Jonathan Brad Chairs. And this was transferred to Northern Arizona University, where I worked in the laboratory of Edwin Lewis. This was after the passing of my father in 2005, so it enabled me to be a little bit closer to my mother during this difficult time. In Flagstaff, I was studying telomeric DNA and porphyrin-based compounds as potential inhibitors. And so the telomere is part of the chromosome that's rich in guanine and cytosine residues, and this essentially causes cancer cells to become immortal. There's a guanine-rich G-quadruplex structure and a complementary cytosine-rich I-motif structure. The guanine residues are non-coding DNA that's added to prevent damage to the genetic material, and this is due to the shortening of DNA with each transcription cycle. That's known as the Hayflick limit, right? And so I constructed an all-atom model of the G-quadruplex or I-motif. This is called the silencer element. And when we stabilize this inhibits the action of telomerase enzyme and adds guanine residues to the cytosine, to the telomere, and so the cancer cells can no longer be immortal. And at the time I did a lot of molecular dynamics simulations, which, well, they were relatively short, maybe a nanosecond or two. I needed faster computers. So this led me to the University of Pittsburgh in the Department of Computational and Systems Biology. I worked in the laboratory of Dr. Daniel Zuckerman, and we worked on a library-based Monte Carlo simulation method of proteins. This LBMC method enabled the generation of a very big protein ensemble structure of the protein, and the ensemble is essentially the collection of that's the 4D representation of the protein's dynamics, right? And so we're looking at the time dependent. We're looking at all the protein fluctuations and dynamics. And so we studied the dynamics of folding and stability of the glucose-galactose binding protein in E. coli. And this is an important component of bacterial chemotaxis which is what helps bacteria find food in their environment. And also in Pittsburgh at this time, I became involved in the University of Pittsburgh Postdoctoral Association. And through that UPPDA, the networking that I gained out of that helped me to find my current faculty position that I have today, right? And so after Pittsburgh, that led me to Oak Ridge National Laboratory, as well as the University of Tennessee, working in the Center for Molecular Biophysics. I worked with Dr. Jerome Baudry and Dr. Jeremy Smith there. And it was there I gained a lot of valuable experience running massively parallel molecular dynamics using NAMD and VMD on some of the fastest supercomputers on the planet. I also studied protein-protein interactions in bacterial chemotaxis, and kind of a serendipitous project in photosynthesis, and I was able to predict the conformational structure of ferredoxin docked into the stromal domain of photosystem 1. This was in collaboration with Dr. Barry Bruce in the BCMB department at the University of Tennessee. And so here I use several interesting protein tools that I've talked about in some of the videos on this channel. One is the protein frustratometer, which is based on energy landscape theory. And this, the other is evolutionary trace, which is kind of like a multi-sequence alignment on steroids. And this generates a ranking of the evolutionary importance of amino acid residues. And together, these two tools can predict the binding sites of proteins, protein-protein interactions, and drug molecules, right? So that was important research. It was good. This actually led me to my current position here in the Department of Chemistry at Tennessee Technological University in Cookeville. Here I teach courses in general organic and biochemistry to nursing students. I also teach general biochemistry 1 and 2, as well as nutritional biochemistry and courses in molecular modeling as well. My research also involves continuing collaborations with researchers at UT and ORNL, and I'm also involved in drug discovery projects with hamigermicin B with Dr. Jesse Carrick, and signal transduction on arrestin proteins and the signal transduction pathway with MKK4 and 7, JUNK3, and ASK1 with Dr. Sean John. I've actually supervised about 8 or 9 master's students and probably about 35 or so undergraduate students. My undergraduate students are actually pretty well known for using, we're probably one of the major users of the high performance computing cluster at Tennessee Tech, which is particularly notable. One of the other major things I'm involved in is 3D printing, and I actually have 3 3D printers at Tech. We've got a Raise Pro 2 3D printer, and I also have 2 Ender Pro 3D printers. So we're fairly involved in 3D printing small molecules, models of small molecules, as well as printing various components for chemistry instrumentation, spectrometers and biochemistry tools and that sort of thing. So if you've seen some of the videos on my YouTube channel, you might have seen several videos related to model trains, and model railroading has kind of been an interest of mine as well. Ever since my uncle gifted us an HO scale model train when we moved to Virginia in middle school. And today I continue to be involved in the Central Tennessee Model Railroaders. Thanks to Dr. Jeff Bowles, another biochemist, for getting me involved in that. So while my professional career involves the modeling of very small things to make them bigger in order to understand how they work, model trains are taking big things and modeling them smaller, studying how they work. And also the kids kind of love it, and it's fun during the holidays and Halloween. There's a lot of cool stuff you can do with model trains. The minute attention to detail in making models of trains and villages is also a skill that I've carried forward in science as well. And this taught me the importance of patience in utilizing all the variables in an effort to make the most accurate model possible. My interest in 3D printing are also very important, not only in printing models of small molecules, but also in printing various model train components and different models of villages and that sort of thing. So that's always kind of fun. And so we're at the end of the year. 2024 is coming to a close. We're rapidly approaching 2025, and I'd say the most significant development in the field of biochemistry is the recent Nobel Prize in Chemistry awarded to David Baker of the University of Washington for his work on protein engineering and design, and John M. Jumper and Demis Hassabis of Google DeepMind for the development of the artificial intelligence software AlphaFold2. And, you know, in the past century, humanity has several significant milestones and revolutions. We go back to the Haber-Bosch process in the early 20th century. This led to the tremendous increase in agricultural productivity. This resulted in a global population increase of from 1.5 billion in 1900 to over 8 billion people today. I'd say the Manhattan Project and the development of the atomic bomb in the 1940s literally changed global politics and launched the world into the use of atomic energy and the atomic age. Of course, we can't forget about the moon landing in 1969 and the Apollo missions. And then there's the invention of the World Wide Web in the 1990s. That literally changed how we communicate across the globe and how science is done as well. And today we're at the genesis of yet another major milestone, and that is artificial intelligence. While ChatGPT is changing the way humans learn and conduct business, making progress to develop at an incredibly rapid rate, AlphaFold2, it actually came out before ChatGPT. This literally solved the problem of protein folding and enabled the prediction of highly accurate protein structures for which scientists can learn more about the mechanisms of biological reactions, design and develop new drugs to treat disease, and study how proteins interact in biological systems. The next decade will certainly be very, very exciting. However, while AlphaFold has made great strides into understanding how proteins fold and predicting their structure, it's not without limitations, and we still have a lot of work to be done. Some might even say that the Nobel Prize was a bit premature. But I don't know, who am I to argue with the Nobel Academy, right? You know, these guys kind of know what they're doing. First, one of the major weaknesses is the limited accuracy of AlphaFold and protein complexes. It works great for simple monomeric proteins, but it struggles with predicting the structure of large multi-protein complexes. It also does not inherently account for these protein-protein interactions in dynamic or transient complexes. AlphaFold also has a relatively poor understanding of protein dynamics, and while it can predict static structures under near equilibrium conditions, this often corresponds to about 1 angstrom of RMSD to X-ray crystallography or cryo-electron microscopy structures. A lot of work still needs to be done to provide insights into protein dynamics and conformational flexibility, as well as the effects of environmental factors like pH, ionic strength, and post-translational modifications. Intrinsically disordered regions also pose a challenge for AI as well. And so these proteins, or they're parts of proteins, they lack a well-defined structure in vivo. And so naturally, AlphaFold really has no idea what to do with these things. But it's a very important concept, especially in signal transduction and regulatory processes. So AlphaFold often predicts them as arbitrary, kind of randomly assigned particles or strings of amino acids. It really doesn't know what to do. And membrane proteins are also highly underrepresented in experimental structural databases. And so this limits AlphaFold's predictive capability with that lipid bilayer environment as well. So that's also important. There's also several limitations in AlphaFold's algorithms in that it only predicts amino acid protein chains, and it neglects things like cofactors, ligands, ions, solvent effects. These are all a critical component of protein function. And so the AI kind of makes an assumption the protein's in an isolated environment, and it ignores all those other things. And ignores things like chaperones and molecular crowding as well. And furthermore, while AlphaFold provides confidence scores in the form of a predicted local distance difference test, that's a PLDDT score, what we lack is a robust framework for calculating uncertainty in the predictions. And so a lot more work is needed here in validating these protein structures, especially for biological accuracy and for the relevance in predictions. AlphaFold also struggles with predicting structural changes as well, and that these structural changes, especially caused by mutations, this can be important in protein engineering and synthetic biology constructs. So more work is also needed to enable the prediction of post-translational modifications such as phosphorylation, glycosylation, and acetylation. And these often have significant effects in biological function. So while AlphaFold predicts structure itself very well, it does not inherently provide information about protein function, dynamics, or interactions. Functional annotations must rely on additional bioinformatics tools or experimental methods. Lastly is the effect of bias on AlphaFold predictions. The AI is trained on existing structural databases, such as the Protein Data Bank. These databases are often skewed towards certain protein families, structural motifs, and organisms. Therefore, its predictions might be focused on specific types of protein motifs, while the actual structure is unrelated due to factors yet undiscovered. So clearly a lot of work still needs to be done over the next few years. Methods that better handle protein-protein and protein-ligand interactions and dynamic complexes are needed. And this is currently being addressed with the next version, AlphaFold2, which is already available. However, AlphaFold2 was released with a bit of controversy. It was initially released as a closed source code and not open source, and this prevented scientists from verifying and reviewing the source code. I think an open source version is now available, so that's good. We also need to integrate molecular dynamic simulations to better capture protein flexibility and conformational states. And that's going to be useful in verifying the results of our models. And this will also help in dealing with the intrinsically disordered proteins and their transitions and different states. The environmental context also must be added to the algorithm, and so we need to include the addition of cofactors, small molecule ligands, other factors such as membranes and pH, ionic strength. This is starting to be addressed, but we still have a long way to go. There's a lot of different things to add. So models also need to be trained to predict structures and include common post-translational modifications and their impacts as well. The bias issue that was previously mentioned with the use of protein databases can also be addressed by incorporating novel experimental structures as well as underrepresented protein families into AlphaFold's training data. And of course, this will improve knowledge of the protein structure and lead to better, more reliable molecules. The additional integration of AlphaFold with experimental techniques such as cryo-electron microscopy, X-ray crystallography, and NMR is also of very insightful use in refining predictions, especially in particularly challenging cases. Especially in comparison with NMR data, for example, this has a unique advantage because NMR is actually an ensemble of experimental structures, and this can be useful in addressing issues with protein dynamics in your AlphaFold predictions as well. So NMR is very useful. The AI can also be adapted for the prediction of non-canonical proteins as well as engineered species and synthetic peptides, and this will be useful in cross-species applications for synthetic biology and drug discovery, for example. Lastly, the development of streamlined tools for biologists with limited computational expertise needs to be addressed to make AlphaFold a lot more accessible. And I give kudos to the University of California at San Francisco's Resource for Biocomputing, Visualization, and Informatics for their integration of AlphaFold2 in the Chimera X software with Google Colab. And they even made a video using cytochrome C as an example that you can check out in my YouTube pages here. And so clearly AlphaFold has contributed significantly to the field of structural biochemistry, especially in the past few years. There's a lot of work that still needs to be done in addressing the limitations of AlphaFold with other computational experimental tools, expanding training data sets, and improving computational models for more complex biological entities. But it's going to be very exciting over the next few years, and I look forward to seeing what we can do, what the future holds. And so this concludes my inaugural podcast of Biochemistry Unfolded, from atoms to algorithms. I hope you enjoyed this topic, and I hope you find it interesting and very useful. If you find this useful, if you find it interesting, please subscribe to the channel, and we'll get more updates to you. Feel free to leave a comment down below and send a like our way, and that will be very helpful as well too. So thank you for watching, and good night. Thank you for watching Biochemistry Unfolded, from atoms to algorithms. If you enjoyed this content, please subscribe to this channel, and we will keep you informed of future episodes. Thank you.